Good day, this is Professor Will, CBMD PhD, case Wednesday, November 20th, 2019 at 16:06, 17 seconds Eastern Standard Time. And today I'm going to be doing a reading on osmolality. So let's get started here. When a microbial cell is in a medium that is hyperosmotic relative to its cytoplasm, the cell tends to lose water by osmosis. Conversely, if the cell is in a medium that is hyperosmotic by comparison to its own fluids, it will accumulate water. In the absence of an appropriate response, there may be growth inhibitory, if not lethal, consequences. Control of internal osmolality is related to other aspects of cell physiology. At low osmolality, the need to preserve internal pH, ionic strength, metabolite concentration, and the concentration of specific ions in a certain range is an important aspect of adaptational response. For example, K+ is the most abundant cation in the cell. It is kept in the range of 100 to 150 mm in the case of Echinacea coli. In contrast, at high osmolality, the need to supplement the cellular milieu with solutes that are at least inhibitory to cellular processes will predominate. Adaptation to osmolality ties near the upper limit of tolerance. Typically also confers enhanced resistance to other stressful conditions such as heat or cold shock. Prokaryotes and other osmoconforming organisms such as plants and vertebrates are generally considered to be close to being isoosmotic with their environment. However, in fact, because of the increased osmolality of the cytoplasm relative to the environment and the resulting inward flow of water, prokaryotic and eukaryotic microorganisms in the cells of many other osmoconforming organisms retain a positive hydrostatic pressure termed turgor pressure, which pushes out against their cell membrane and cell walls. In response to turgor pressure, the prokaryotic cell wall expands elastically until its tension exerts an opposing force equal to the hydrostatic pressure. The pressure inside gram-negative bacterial cells is on the order of 2 atmospheres, 30 pounds per square inch, and can reach up to about 20 atmospheres in the case of some gram-positive bacteria. Measurements of bacterial turgor pressure indicate that it does not vary much during cell division or during the various phases of growth. It has been proposed that turgor pressure is maintained within narrow bounds in order to produce the necessary mechanical stress for cell wall expansion. This then could be one of the reasons why it is important for cells to accumulate osmolytes as they grow and divide. In describing the osmotic adaptations of microorganisms, another important term is water activity, abbreviated A sub W. This is the ratio of vapor pressure of water in equilibrium with an aqueous solution divided by the vapor pressure at the same temperature of pure water. The values of A sub W vary between 0 and 1. Microbial life has evolved widely different preferences for A sub W and the amount of change in A sub W that can be tolerated. Thus, osmotic stress is different to different prokaryotes. E. coli typically contains 70% of its cytoplasm as water, although this value may vary by close to twofold depending on the osmolality of its environment. Bacteria such as certain species of Caulobacter and Spirillum can grow at A sub W close to 1.000 pure water. Marine bacteria adapted to the A sub W of seawater 0.980. Some members of the archaea can grow at A sub W down to 0.750, and certain fungi can still grow at A sub W as low as 0.700. Similarly, endolithic lichen and bacteria of deserts must be adapted to extremely low A sub W value. The bacterial species Vibrio costicola, as well as the bacteria family Halomonadaceae, are particularly fascinating because of the tremendous range of A sub W they will tolerate. They will grow over a range of water activities between 0.98 close to fresh water to 0.86 close to saturated NaCl. Decreasing water activity by freeze-drying or salting is one of the principal means of preventing food spoilage by microorganisms. Water potential, a related energy term that is expressed in units of pressure, is also used to describe the concentration of water in some scientific fields. Organisms that can grow in media of high and average solutes concentrations are called osmotolerant, and those that actually prefer elevated osmolalities are termed osmophile. In the environment, osmotic adaptations are usually correlated with the presence of particular solutes. Because of this, it is important to distinguish general osmotic requirements from those regarding specific chemicals. Marine microorganisms, for example, typically have a specific need for a certain amount of sodium ions in addition to a moderately increased osmolality over their terrestrial microbial counterpart. They are thus moderate halophiles. Certain members of archaea domains such as Halophorax and Halobium species have still higher requirements for sodium ions and are described as extreme halophiles. In terms of salt concentration enabling growth, microorganisms may grow from close to 0 m to close saturating concentrations at 5.2 m sodium chloride Saline Vibrio, Causticola, and bacteria family Polonidaceae are described as haloversatile. Desiccation results in extreme osmotic stress and low A sub W values. Some microbes die almost instantly in air, whereas the spores of algae, fungi, and bacteria in the cyst protozoa are very resistant to dying. The vegetative cells of many gram-positive cells and bacteria of the family Dinococcaceae are also quite resistant to desiccation. In the former case, this is believed to stem in part from their thick cell walls, which may help protect membrane integrity. Protection of damage to chromosomal DNA during dying is also critical for cell survival. The extremely efficient DNA repair capabilities of Dinococcaceae have been found to be required for their desiccation resistance. The focus here is on the physiological processes by which microorganisms adapt to osmotic stress. However, note that one of the most important responses of motile microorganisms to change in osmolality is to swim away into a new form of favorable osmotic environment. This process is known as osmotaxis. Bacteria repelled by pure water are attracted to their particular optimal osmolality and repelled again by supraoptimal osmolality. By moving back into a region of optimal osmolality, the need for substantial physiological modification can be greatly diminished. However, it appears to be mediated by a direct effect on the rotation switch at the base of the flagellum. Chemoreceptor proteins in the cell membrane also help cells adjust their motility in response to particular solutes such as NaCl. Effects of increased osmolality on proteins. During exposure to hyperosmotic stress conditions, solutes will increase in concentration inside cells. Many of the deleterious effects on hyperosmotic stress can be interpreted in the context of solute effects on macromolecules. Increased osmolality tends to promote the dehydration of proteins. In a sense, proteins can be considered as behaving similar to a semi-permeable membrane. The exposure of a protein to an environment in which the A sub W is lower than that of the water bound up to the protein will result in the release of the bound water to the surrounding solution. One line of evidence indicating the profound influence of osmolality on protein structure has come from the isolation of osmoremedial mutants. These microorganisms encode a mutant protein that is non-functional when the cells are grown in media of low ionic strength, but that regains at least partial function when the cells are grown in media of higher osmolality. Osmoremedial mutations have been identified in genes encoding both soluble and membrane proteins of monomeric or multimeric configuration. Most, if not all, temperature conditional mutants are also osmoremedial. Different solutes can have different effects on proteins and their structure and activity K+, and glutamate are important intercellular osmolytes at low concentration, but as do other ions, will inhibit many enzymes when present at high concentrations. Solutes that carry a net electric charge are generally more damaging to protein stability than nonpolar or zwitterionic solute. The charged solutes can be further subdivided according to the Hofmeister series, also called the lytrophic series, according to their effects on protein. Stabilizing ions do not bind strongly to proteins or interfere with the water molecules coating the protein surface. These ions tend to preserve the native protein structure. Thus, stabilizing solutes distribute themselves away from the protein, which is preferentially hydrated near its surface. Because the exclusion of stabilizing solutes, molecules from the water around the protein, reduces the entropy of the system, is accompanied by an energetic cost. This cost can be reduced by minimizing the area on the surface of the protein that is exposed to solvent, thus resulting in a more compact protein structure. Thus, even in the presence of stabilizing solutes, in the absence of protein modification, increased ionic strength will alter protein structure. The Hofmeister series does not apply to all proteins. An exception are the proteins of some of the extremely halophilic archaea. Proteins isolated from these organisms are stable only under conditions of high ionic strength. Proteins isolated from these organisms are stable only under conditions of high ionic strength. Indeed, the interior of archaea of the family of Halobacteriaceae may accumulate more than 5 mkcl. Their proteins have an excess of acidic over basic residues when compared to their corresponding non-halophilic protein. Because of this acidic character, these proteins are able to fold correctly only in the presence of sufficient counterions, such as K+. High potassium levels may also balance the amount of water bound to proteins in the burial of weakly hydrophobic residues, such as alanine and glycine. Instead of passive cell volume regulation, prokaryotes respond to osmotic increase by selectively increasing the concentration of certain solutes, thus reducing water activity but returning cell volume and turgor pressure to approximately Prestris level. The exposure of E. coli cells to hyperosmotic stress results in a sequential series of adaptive steps. The first line of defense is a large increase in the rate of potassium K+ uptake. This appears to be necessary for the resumption of growth. of the plasmalized cell. Subsequently, the synthesis of glutamate is induced. This charged amino acid serves as a principal counter-ion for K+. In some organisms, e.g. Salmonella typhimurium, the accumulation of glutamate is necessary for the optimal growth of media of high osmolality. However, because glutamate is a substrate for a variety of cellular enzymes, specifically a key intermediate in nitrogen metabolism and a component of proteins, its concentration in cells may be regulated. With the uptake or synthesis of compatible solutes, K+ levels in E. coli decrease as a result of efflux and glutamate levels decrease as a result of turnover. The synthesis of tree halose results in the disaccharide eventually replacing K+ and glutamate as the major osmolyte inside the cell. Mutants impaired in K+ uptake produce tree halose more rapidly, and mutants unable to synthesize tree halose exhibit increased intracellular K+ concentrations. Thus, E. coli and presumably many other bacteria have evolved a signaling process for communicating and controlling the levels of the osmolytes K+ and tree halose together inside the cell. K+ is believed to act as a second messenger that influences not just tree halose levels, but many of the later steps of the cellular response to hyperosmotic stress. Consistent with the hypothesis, there is a dependency on the external K+ for osmoal adaptation in the absence of external capable solute. Good day, this is Professor Will, CBMD, PhD. Today is Wednesday, November 20th, 2019. It is 7:59:51 seconds Eastern Standard Time. And this concludes my reading on osmotic stress. Hope you enjoyed listening. Have a great day.